Welcome to my keynote on in vitro neural stimulations. And I thank you for the organizers for asking me to do it. And I guess when they asked me to do it, they really wanted to maybe bring something different for this brain stimulation conference. So my topic today is stimulation assessing emerging in vitro neural models. So just to give a little bit background, what my group is doing at Tampere University. So my group is kind of having three different main cell targets. One is epithelium, one is neurons and astrocytes, that is today's topic, and third one is cardiomyocytes. And what we do is three different things: in vitro methods for electrophysiology especially, and 3D imaging, especially tomographic imaging on mesoscopic or cellular systems, and computational modeling and analysis of all these. And of course, today's topic is neurons and astrocytes. And I'd like to also introduce two projects that I'm involved. One is Finnish Academy Center of Excellence on body-on-chip research. And it kind of spans from epithelia, especially neurons, but also cardiomyocytes, but other tissue types. And another one is HERMES, that is a hybrid enhanced precision medicine system, and that is a European future emerging technology project. I introduced this here because you will see some slides with these logos, so you know what these mean. Okay, so let's look a little bit what I'm going to talk to you. First, we'll look a little bit about in vitro stimulation neural models. So why we need to have these models. And then we look a little bit about the engineering behind these, because actually there's quite a lot of different engineering. And then we look at the biology side. So what kind of models we can have in vitro, and a little bit what are the kind of challenges and what kind of stimulus system we can have there. A couple of examples. And then future directions. So another one is really looking how we can kind of improve the biomimicking complex systems. Another one is really looking on this in vitro in vivo translation. Okay, so what is in vitro neurotechnology? Is it a brain on test tube? Could you call it brain on a dish? And actually, it's more or less like this. In the brain stimulation community, and most of you are, what components you have? Of course, you have the brain. You stimulate the brain with, anyway, many times with electric and magnetic fields. Then, of course, you have the recording system. You record the responses. Many times it's EEG or MEG, among other things. And then you have the signals. And in principle, in vitro system have the same. We have the recording system that is called here microelectrode. And this is actually something I would like to concentrate more on this talk. Then we have the brain, and that is a neural system that lives on top of these electrodes. And then we have the signals. So very much the same components. And of course, this is about modeling. So why we do these in vitro models is we would like to figure out what the neurons are doing. And of course, it's a model of the real neural systems, a way or another. And of course, one key question is that how good are these models? So if you think in general, how we model neural systems, of course, we can have the human brain, but of course, it's somehow limited what we can do with the human brain. Normally, we are quite limited to the outer surface or scalp. In mouse and other animal models, of course, we can go in and do some more invasive studies as well. But many times we would like to go even closer to neurons. And that's why it's sometimes much better to go on in vitro systems. So culturing the neurons in vitro. Of course, here you can have different kind of scales. A lot of studies have done on kind of single cells, couple of cells, where we look the like patch clamp electrodes, the kind of ion channels, or then here it's more like network level. And I think this is kind of interesting level because it's the kind of more relation to the brain, because here it's a network that has also very many similar properties of the brain. Even it's, as said, just a model. Okay, why we do it as well is kind of translation to humans, because many of the work done, closer look what neurons do is done in animal models. If you would like to see, for example, drug effects or some other effects of the neural system behavior, you would like to go to more like humans. And in vitro offers this way, because we can actually have the in vitro models based on human cells. I'll talk that a little bit later. So what are targets of electrical stimulation in vitro? There's actually quite a lot of different targets. Starting from cell differentiation, we can think about it if we can boost electric machinery on kind of early phase neurons or stem cells. Those could maybe differentiate better the neurons. That has been done. Also, we could think that we could have a better neural growth. For example, we could try to grow axons. That could be helpful in certain diseases as well, and test it in vitro. Sometimes it's also nice to actually look what are the adverse effects of stimulus, because there are many. Of course, we can look the stimulus to look the activation, information propagation network. We can challenge the network communication. These are as well the same things done in vivo, like human brain stimulation wells. And of course, we could try to do also network modulation and look like learning events in networks. Then, of course, if we can, of course, learn a lot of things in vitro that we can hopefully translate to in vivo, or build this future bio-ICT hybrids where we could kind of, let's say, have electronics and neurons living together, whether it's outside the human body or even implanted. I have a couple of examples of most of these areas. So what is the technology behind these in vitro systems? First of all, as I showed before, these are the components. We have the same ones that are in vivo, the cells, the brain, the measurement system, and the signals. But of course, there are some similarities and differences. And so those are the same things. Electronics, electrodes to record. We have a brain or the neurons. We have the signals we would like to analyze. And then also in vitro, we have structural and functional measurements. Similarly, as in vivo, we have MRI, functional MRI. Here we have imaging methods, microscopy methods, for example, to stain certain neural structures, or then functional imaging like calcium imaging. And of course, then we can analyze these systems in a very similar way as is done in EEG, or also looking at the connectivity on the brain on MRI. But of course, there are differences. One major one is that, of course, if you do human or animal studies, you have your culturing system. You have your animal or your patient that is actually taking care of the brain. But in vitro, of course, you have to build your culturing system and environment. One thing is that in vitro, we don't have any other means to stimulate the system. In vivo, of course, you can use different sounds, all kinds of senses to alter the brain functions. But here, there are none of that. Then we have only part of the brain or part of the neural system. So there's no higher kind of cognition available. And similarly, there's no response. The functional responses are not as in vivo, not kind of, let's say, breathing or heartbeat change, nothing like that. Or there's no locomotion or something else. So all the analysis is based on signals and, of course, the images. And this is just a basic tool that many uses in vitro. That is the microelectrode array. So these are maybe half a dozen manufacturers around the world to make these systems. They are more or less the same, the basic form. So we have this kind of quite sizable dish where you can culture cells. And then we have bottom there electrodes, like here. The electrode distance is 200 microns, that is quite typical. It can vary quite a lot. And also the electrode distance here is 30 micron, and it can range from a couple of microns to, let's say, 100 micron. So they're pretty small. So they are microelectrode arrays. And of course, one important component I'm not going to talk today very much is the electrode itself. Like here is titanium nitride surface that makes this kind of large surface area, almost like a tree structure. But that is very important if you would like to stimulate your cells to have very good current injection capability. And of course, there you have your network growing and you record them. And if you would like to stimulate these networks, normally it's very similar kind of pulses that you deliver, for example, DPS. It's biphasic pulses many times. And those are like average kind of signals that we can use, couple of milliseconds or parts, microsecond pulses, half a volts, 1 volt pulses. And then you get the responses. Not all MEAs are similar. This is another type that's kind of interesting. It's like building growing your cells on top of CMOS circuit. So this is actually CMOS circuit, and on the right, the small dots or round ones are electrodes. They're actually a gate of a FAT transistor that you can grow your cells on top of those, and they record the charge transfer from your cell surface. And there's also quite a number of stimulus sites. So these give quite a lot of different kind of application areas as well. Much more detailed picture, but a lot of work has been done in the basic MEA. And if you think about the microelectrode array signals, it's quite different than EEG, because now we are really recording from the neurons, but it's still field potential. So what you get here is a signal that on the right, and you can see this looking like a noise, and then there are a couple of spikes emerging, and those are the neural signals from the really adjacent electrodes. Normally we can record something like signals around 3 times the area of the electrodes. There are no volume conductions as just same way as in brain or EEG. So it's much smaller electrode area. And then if you think about this signal, the spikes we actually measure, and then or we identify them normally very simple way by having a kind of cutoff voltage thresholding. And sometimes the spikes form this kind of spike trains, or even very dense spike trains, and that are called bursts. And many times the analysis of these signals are based on statistics, how much there are spikes and bursts, and what is the transition between spike and bursts. And this is how they look when you identify the spikes and mark the spikes every time on all channels. There are 60 channels, and you can see these dense groups where the spikes and bursts are, and you can see that they are in every channel time to time. That means that the whole network has synchronous spikes or bursts. So we analyze spikes and bursts. That is the basic thing. That the statistics of those. You can look more carefully. You can actually also look the burst analysis and sort the spikes. Like here, when you look more carefully in one spike, they actually differ. Then you can classify your spikes to different forms, and then look how many of those spikes are different forms, and how your stimulus affect them, how the drug, for example, affect these spikes, and how much they are in maybe spikes, and how much different kind of groups are in bursts. Of course, you can also look the spatial distribution of spikes or bursts in your network. And of course, one same kind of thing that you do on the brain network, we can also analyze the connectivity inside these networks. This is one example of those methods that we use based on spectral entropy. But there are many methods for this, similarly as in EEG. And it has exactly the same purpose, to look the connectivity and look the kind of how it's developed over time, for example, here. In vitro is kind of nice because we can do a lot of things. One is that we can, like here, manipulate the cells. We can inject materials, drugs, or genes to the cells if we like. We can, of course, measure them. We can culture the neurons on top of these electrodes. We can have mechanical stimulation. We can have heat distribution. We can bring gases on this system, or we can have fluid shear, for example. This, by the way, slide of my colleague Pasi Kallio, who is mostly doing these in vitro systems for microfluidics. And of course, we can then measure all kinds of things like patch clamping, microelectrode. We can even look the migration of cells, or like here, cardiomyocyte speeding. So the image-based analysis, or we can even have kind of environmental sensors. So in vitro system is kind of nice because we can really look how different kind of methods, how to model really biological system and look the environment, have different diseases in vitro on this, maybe build trauma, have different drugs, and maybe also some phenotypes, and really look the mechanism of things much more carefully than maybe on in vivo systems. Okay, so what is the kind of some of the main challenges? Of course, there are a lot of kind of engineering challenges to do it, but partially engineering challenges are linked to the biology. And of course, the main question is how we can make these in vitro systems better, how they can mimic better the brain structures. And of course, there's quite a lot of ways to do it. I'll look some of them. One is really to go from 2D to 3D. But of course, another direction is that how to make these structures more defined and repetitive. If you think you're culturing cells on vivo, they actually look very different every time you culture them. There are ways to do it. So this is just a kind of little introduction on what kind of models we have in vitro, because we can have different kind of models. One is ex vivo tissues, another one is culture of dissociated neuron cells, and then really kind of strong emerging area, stem cell differentiated neurons that has been around like last 10 years. This is just an example of work that really started the in vitro system, and that is the ex vivo brain slices. Like here is a rat hippocampal slice, and because there's an injury in this hippocampus, it's also prone to epilepsy. So you can see at the end on the recording how the spiking turns to the more like epileptic bursts kind of activity. And these slice recordings have been a lot of also stimulus recordings where the connectivity of the slices of brain, parts of brain has been studied. Also, a lot of work has been done in the retina because this is an excellent tool for looking at the retinal circuitry. Okay, and then very common is animal dissociated neural cell cultures. What it means is that you have really brain cells from living animals, and then you really kind of redistribute them on the surface of the mare. And for example, this comes out of that. So they regroup, grow these kind of mini areas and connect connections. These are pretty easy because they're pretty fast. A couple of days and you can have this kind of nice culture and start getting signals. And of course, one interesting what we talked already is the human cell cultures. To my knowledge, this is one of the really first papers to show that we can grow human stem cell based neurons on MEA, and we can get nice signals, and they generate also network. And this just shows as well that when you start from stem cells or precursor cells, it's very slow. They start to make connection, a couple of weeks takes, and they have a couple of spikes, and it takes many weeks to get this kind of level of integrity that you can get bursts like in slices. But of course, what really changed the whole thing, these were embryonic stem cells, was the emerging of pluripotent stem cells, and these really changed the world, because now some of the ethical issues were cleared, but also the idea is that we can have cells from any of us. So they're really patient cells that can have diseased cells, because if the patient have some diseased, or then of course we can think about they are excellent for personalized medicine. I think many of you know this, so I don't take more time on these, but these are really available quite widely. You can buy these cells from the shop as well. Okay, but let's look a little bit about this. So these are dissociated rat neurons, and you can see a nice network, some connectivity, not the signals here, but of course the signal as well. But what is kind of bad here, that whether you have human cells or rat cells, every time you plate the cells, they form different networks. So they don't have rules actually how to make the networks. They don't make brain-like structures. And that's why people have tried to really make systems that better mimic the brain, or really look how to make these more kind of defined. And one is really to build this kind of environment where you have kind of network structure emerges. Like you have something like material wells that force the cells to form patterns. That has been used a lot, or some other patterning methods on the surface that guides the neurons to go certain directions. Don't go very much in detail here, but I welcome you to read, for example, this paper that is from Janos Vörö's group. That they made the nice patterning, and also were able to make this kind of system where the neurons are making directional connections, not just connections on a certain kind of way, but also directional connectivity. So of course, this increased repeatability and control, but it doesn't necessarily bring kind of more biomimetic systems. This actually one attempt from Tampere, my colleagues Susanna Narkilahti and Pasi Kallio, how they made this kind of microfluidic system. On the left, you can see that there are two chambers, the blue ones, there are neurons growing. And then on the middle, the neural cultures actually make connection, they have actions. And there's a third chamber where they grow oligodendrocytes. And oligodendrocytes are cells that make then myelinization. And of course, this is much more like brain structure, because you have like two brain areas, and then they are connected by myelinated cells, just like the white matter tracks in brain. So this is the step forward for that direction. And of course, here the stimulation was made easy. You can stimulate another side, and then you can follow how, when the pulses reach another side and go through the network or the tunnels with oligodendrocytes, and you can follow the myelinization by speeding up the travel of the pulses. Of course, another way to going is really looking at 3D cultures, because brain, after all, is not growing on glass, not even inside glass, but still it's 3D, and neurons and astrocytes, other cells, really would like to be on 3D and not in 2D. This actually old slide from, we had this kind of 3D neuron EU Future Emerging Technology project many years ago, where we actually growed, this is again from Susanna Narkilahti lab, it's kind of neural cultures that start to emerge and 3D hydrocells. And that is the famous, most kind of used material for scaffolding on 3D neurocultures. What is now emerging actually is this kind of organoid structures, and these are structures that kind of neurons, with treated properly, they start to make a more like brain-like structures and starting to organize themselves. Not really exactly like cortex, but at least much more organized way than the other structures. And this is from our Hermes project from Ilaria Decimo group from Italy. And left you can see the neurons on the map 2, and GFAP marks the astrocytes. So you can see some organization there, and the structure was about to mimic the hippocampus. So this is the kind of how it looks in 3D. So it is some resemblance actually. So these kind of models are also emerging. Not very much studies on stimulation yet though. This is just one example we did in collaboration with University of Auckland and University of Wollongong, Australia, and on 3D. An idea here was to build this kind of 3D electroarray that you can see on the right. This is from the Auckland site. And then try to kind of look how this electrical stimulation given through these electrodes kind of drive or boost this network on top that are made from human iPS stem cell derived neural cells. And here we didn't look the activation or how the neurons are activated, because this is early network. So here you look actually the functional imaging, and that is how the networks start to evolve on top of these stimulated electrodes. And clearly the stimulation enhance the kind of growth of actions and enhance the network kind of construction. Just an example of kind of what you can do in vitro as well. So there's a lot of work actually done, interesting work on so-called biohybrids, where we can have kind of combination of biological systems and ICT. Of course, the whole MEA system is like that, but looking more like functional things. And this interesting paper is quite many years old, but still it's kind of interesting approach where they hooked up this MEA system, a robot with sensors, and then they put it on, and the control on the robot movement from the neural system and the maze. Very interesting story. It was a little bit still like a toy system, because there was not really learning, but it was more like selection on input and output channels from the MEA that provided this kind of logic for this, let's say, from sensors to motor. But this just shows the kind of interesting area that you can go as well. What we have done recently is kind of looking the kind of idea that how we can have closed loop on-demand based stimulation in vitro. I would have hoped to give you actually a little bit more on this, but present situation have kind of, let's say, stopped some of our experiment on lab, and this is based on our early paper that was published last year in a conference. So if you look at the MEA systems, you have the array, then you have the head state amplifier, interface board, and PC software at the end. Normally the stimulus, you decide what the stimulus is, you drive it, and then you analyze it later what happens. Actually, what is nice thing is that the interface board has a digital signal processing that actually could offer real-time analysis. So how about to drive this interface board and build an adaptive stimulus system that stimulate only when there's some condition happening on the signaling of the neurons on the MEA system. So I have this kind of closed loop system that neurons itself kind of tell when to stimulate, or at least there's a logic behind. So what actually we did first was very kind of simple scenario. And this is really an idea that we record from one channel of the MEA, and then select another channel to stimulation. And of course, we look from the microscope imaging. There's a kind of connectivity between these, and also there's a synchronous activity between these cells. And when something happens on those recording electrode, we start to stimulate. And this is really kind of idea very simple. If there are kind of mini bursts or 3 spikes in 500 milliseconds in our observed electrode, then we start to have 5 basic pulses, 100 pulses actually on our receiving electrode. Of course, these are like the signal they look like. This is a spontaneous activity, the spikes, and then you have how the spike look like. And when you stimulate, you can have huge signal there, because they are normal are like tens of millivolts, and then this is like a volt signal, and then you can see the response on different channels. Unfortunately, this response is not necessarily kind of straightforward, because these are really random dissociated cultures. But when you look at the statistics, something will happen. But let's look actually first what could happen also on the electrodes. So on the left, you can see the original, let's say, typical neural structures. So that the green ones again are the neuron system, and the red ones are glia cells, and blues are cellsomas. On the right, it's after stimulation. You can see that the electrode black dot on the center, they are not really green very much around it. There's green, but it's very much blended with the glia cells. So there's definitely a change after stimulation there. And this is what happens in vivo as well when you have implanted electrodes. When we now look at the average spike and counts on what happens there. So this is a baseline before stimulation. And you can see there that we use this stimulation on like a 20 days or long, kind of many days stimulation as well, and then there's a post stimulation at 20 days. So this is before the stimulation, then after the stimulation. So definitely the functional stimulated cells, feedback stimulated, they had more activity than the predeterminate stimulation. And also it remains on post stimulation. And same thing happens on bursting. So this is how when the neurons are these spike trains. So the functional stimulation clearly provided kind of different feedback than the normal stimulation. And of course, this is very preliminary, but it seems that the effect of stimulation neural system depends on previous state of the system as well. And maybe, hopefully this brings something interesting in our later studies. Okay, think about future, where we go. I kind of introduced 2 of our projects we are involving. Maybe it's kind of some idea. This is our Center of Excellence in Body on Chip research. And as said, we have 3 groups from different biology. So they represent different kind of cell types. And then we have people from kind of engineering side, micro nanosystems, biomaterials, biophysics and imaging, like myself. And what we do here is really environments for the cells to grow, but not just cells. We would like to do a system that are tissues, because in a way, like neurons are not the only type of cells in the brain. There are a lot of different types of cells. And cardiomyocytes are not only cells in heart. So you have to think what is the real tissue. And then, of course, every tissue have innervation, every tissue have vascularization. How to build those in these in vitro systems that mimic the body. So this is what we have done already. So we have environments we can grow, we can follow them optically and electrically by these MEA systems, and control all the environment for most environmental factors on these systems. And this kind of early kind of idea prototype where we are heading. So really looking for this kind of multi-compartment chip. And idea is that there should be a common vasculature and innervation going from one chip to another. And of course, this enables not just to look at the input-output of neurons, but looking like systemic effects. What is the effect of stroke? Some metabolic challenges like oxygen deprivation. What happens there? And of course, this is the idea that we would like to go is more like diversity, kind of personalized medicine. How these can be used for testing drugs or looking diseases, personalized diagnostics and drug discovery. Another project that we are involved is this kind of future emerging technology project in EU called HERMES, as I told before. An idea there is to heal the brain disorders with tissue transplant, but not just like to have the tissue, or not just like to have a neuroprosthetic like electrodes to stimulate something, but really idea is to put these together so that we actually have implanted cells, and then also this neuroprosthetic idea. And of course, it means a lot of different kind of areas to study. So we have in vitro cultures of organoids for the graph. We have in vitro and in vivo models of animal models for epilepsy. We have a lot of work in stimulation in vitro and in vivo. We have to develop new methods for assessing neural dynamics, look into artificial intelligence control, and develop this kind of implantable neuromorphic system with electronic CMOS and MEMS. But this is a future emerging technology project, so this is interesting work, and of course 4 interesting years ahead. Okay, so this is kind of concluding part, and I would like to say that these kind of in vivo, ex vivo slice in vitro, that means, has had already a lot of impact on neural research. But now these new, let's say, defined neural culturing systems are emerging, especially using human stem cell-based system. And both directions are developed now to really better control the neural system on chip, and also to improve the biometric system. And these will have potential for another direction is personalized medicine, really novel tools for systemic effects. Another direction is looking these for this bio-ICT interactions and how to make the transfer from these in vitro systems to in vivo. I'd like to thank my lab, and especially those people who are involved in electrophysiology and doing the neural work, Jarno Tanskanen and Annika Ahtiainen, especially because some of the material was here in this project, and our collaborator Susanna Narkilahti. We have done a lot of work in neural side, and our human center of excellence work there as well, and of course the HERMES consortium. And thank you for listening. I hope you enjoyed the talk and got interested also the translation from in vivo brain stimulation to in vitro. And there you can find also some of the let's say links to our work as well. Thank you for listening.